Today what we're going to talk about is we're going to talk about DNA repair and start out with what happens when the information to the genome changes. The information in the genome is encoded in form of sequence, right? Is the sequence of A's and T's and G's and C's, and sometimes this sequence can change due to the mutations that occur on the DNA. So introduction of changes to DNA sequence is basically what we call mutation. All right. Mutation through DNA damage, so that's damage to our DNA, occurs daily all the time. Okay? So estimates of DNA damage per cell is in the range of 10,000 to 1 million a day. So that means our DNA is constantly damaged. Okay? This damage could be due to inherent instability of the DNA itself. For example, sometimes the bond connecting the purine to the sugar gets hydrolyzed, and that leaves a base with the sugar but without the purine, right? So that leaves, then there will be a spot with no information, no ATGC information. So if this is not corrected, if it's not repaired and gets replicated, DNA polymerase deals with this by incorporating a random base. So you're going to have a change of information if the wrong base is inserted. Normally, the cellular reactions in our cells that happen in the mitochondria, some of these reactions, as a byproduct of ATP production, creates reactive oxygen species, which can also damage both the mitochondrial genome and the nuclear genome. So actually, there is an enzyme called peroxidase dismutase that works as an antioxidant defense, as the first defense to prevent the damage. But if the damage occurs, there are repair mechanisms, which we're going to talk about today, repair that damage. Okay? And I don't remember why I put this picture. It looks cool. All right. So what are the types of different damages that can occur? Here's a table that summarizes all of these. Okay? It's a little bit of a boring table, but please pay attention and learn what these type of damages are and what kind of repair mechanisms can repair them. All right, so the DNA damaging agents outside the cell, so these are not inherent in the DNA, right? So these are damages coming from outside the cells, could be ionizing radiations, X-rays, and anti-tumor drugs. The types of lesions that these kind of damaging agents make are double-strand breaks, single-strand breaks, and intrastrand and interstrand crosslinks. So the type of repair mechanisms that would repair these are non-homologous end joining that would repair double-strand breaks, and homologous recombination that could repair double-strand breaks, as well as single-strand breaks. So the UV light and some chemicals can cause addition of bulky adducts. That means like addition of extra molecules to the bases in the DNA. And the pyrimidine, formation of pyrimidine dimers, these are crosslinks between the T's. And these are repaired by nucleotide excision repair. The other types of damaging agents, such as oxygen radicals, hydrolysis, and alkylating agents, can change the chemical structure of the bases. They can leave abasic sites. These are sites where the DNA sugar phosphate backbone is intact, but it lacks the purine or pyrimidine. It can cause single-strand breaks and 8-oxoguanine lesions, and these are repaired by the base excision repair machinery. And some replication errors, these are errors made by the DNA polymerase, can cause mismatched bases, insertions or deletions, which are typically repaired by mismatch repair. So this column here, fidelity, that means how well do you restore the original information through these types of repair mechanisms. While these nucleotide excision, base excision repairs, and mismatch repair have very high fidelity, so they restore the information completely, the non-homologous end joining does not do a good job. It can repair the double-strand break, but can make lots of mistakes. And the homologous recombination is usually high fidelity unless something unusual occurs. All right. So let's classify now the type of damages into two groups to go through them. So some of the spontaneous errors in replication, like a collapsed fork, mismatches, and exposure to various mutagens can cause mismatches, nicks, adduct, and these are repaired by mechanisms that can repair these. And then there are mechanisms that can repair double-strand breaks. All right, let's start with the nicks. When there's a nick in the DNA, a nick means it's a cut in the sugar phosphate bond of one of the two strands. Okay, so when there's a nick in the DNA, and if the cell is replicating, what's going to happen is that, for example, this cell is replicating and replicating through the leading, it's a leading strand, and there's a nick in this leading strand. What you're going to have is the replication machinery coming up to the DNA nick, but it's not going to be able to pass through the DNA nick because the template is broken. And what that leaves you is it leaves you a double-stranded end in one of the sister chromatids, right? So what happens is that this double-stranded end is recognized by homologous repair machinery. You've learned homologous recombination. And what happens is that an exonuclease chews back one of the strands backwards. This is a 5' exonuclease, so it's going to chew back the 5' end of the double-stranded DNA, leaving a single-stranded DNA out. So this single-stranded DNA is going to look for homology, and where it's going to find is it's going to find it right next to itself, which is the other sister chromatid, which is doing okay, right? So what it's going to do is it's going to invade the sister chromatid, and through that invasion, through RAC-A and RAD51, this branch is going to migrate. So that is what we call the holiday structure. And the migration will continue until it's going to be where the holiday structure is resolved. So now what you end up is the newly synthesized strand that's continuous and a couple of nicks in the other strands. These nicks are going to be repaired by ligases that are going to put through together. And now you basically restored the replication fork in which there is a leading strand and you start synthesizing the lagging strands on the other end. Does that make sense? All right. That's how the replication coupled homologous recombination can take care of these kind of nick problems. All right. Another common problem that occur in the cells is the deamination of the methyl cytosine base. In a lot of organisms, the cytosine base is modified. What that is that there is an additional methyl group is added to the cytosine. Okay, so that's what we call 5-methyl-cytosine. So the methylation is added to the 5th carbon. So if this base is deaminated, it leaves a base that looks very much like thymine. It's exactly like thymine. Okay, so if you have 5-methylcytosine in the genome and this gets deaminated, you're going to generate a thymine. So if that happens in a normal double-stranded DNA molecule where you have usually had the C against the G, right? And this is methylated, and it goes through deamination, and it creates a thymine and gene. Now, if this gets repaired back to the original form, no problem. But sometimes it doesn't get repaired in time, and if that happens, and this double-stranded DNA goes through replication, these strands are going to be separated, and the replication machinery is going to put an A base against the T in one of the sister chromatids, and it's going to put a C against the G. So what you can see here is that one of the cells is going to inherit this chromosome where the sequence of the sequence has changed, while the other cell is going to inherit the wild type sequence. Does this make sense?
In terms of when the cellular machinery has to fix the mismatch, how does it recognize which one is correct and which one is incorrect?
We're going to talk about that just in 1 minute. All right, how does this work, right? So the machinery that does base excision repair is called base excision repair machinery. It repairs damaged bases, okay? So there's a whole bunch of DNA glycosylases that are specific for different sets of chemically modified DNA bases. So there is a glycosylase for oxyguanine, and there is a glycosylase that will recognize A basic sites. These are sites that have the sugar phosphate bond but doesn't have the purine or pyrimidine. And what they're going to do is a DNA glycosylase recognizes is evolved to recognize this problem, where it's going to see a thymine against a guanine. Okay? Because this is such a common change that occurs, DNA glycosylases and the machinery has evolved to assume, doesn't know for sure, that the T is the wrong base. Okay? So what it's going to do is it's going to change the T into a C. So the first step in this process is the DNA glycosylase removes the base. So it leaves an A basic site. So this A basic site is then recruits the APE1 endonuclease, which cuts both sides of the DNA sugar phosphate. It cuts one side of the sugar phosphate backbone, and another protein called AP lyase is going to remove this completely. So what you're going to have is you're going to have a single nucleotide that's missing, right, in the double-stranded DNA. That single nucleotide that's missing gets filled in by a special DNA polymerase called DNA polymerase beta that fills up this gap, and the phosphate backbone is ligated by the DNA ligase, and so this wild-type DNA information is restored because DNA pol beta is always going to insert a C. Does that answer your question? Any questions about the mechanism?
What's like the necessity to remove the entire base? Like, why can't they just add T?
If there was an enzyme in our cells that could do that, perhaps that would be another way to do it, right? But we don't have that, right? So the solution is then remove the whole thing and then let the polymerase machinery to fix it. Any other questions?
Are there not like problems caused by assuming that the T is the mistake?
It could, right? If this assumption were not true, there would be a problem. But most of the time, this is such a common mistake that is made, it's going to be correct. Right? Any question? Okay.
I was asking, does the T become the C after deamination?
T become a C after, no. The methylated C become a T after deamination. All right. Which one is the wrong strand? We answered that question already. Okay, so this is the type of damages usually introduced by inherent instability of the DNA and some chemicals. But there are a lot of mistakes that are made by polymerase itself as well while it's replicating. Now, polymerases are amazing enzymes. They're so accurate, it's unbelievable, right? They're doing a very complex reaction, and they're always getting it right. But sometimes they don't get it right. And there are polymerase mechanisms that have evolved to correct these kind of problems. One is the polymerases that have high fidelity. That means they are very true to replicating correctly. One of the mechanisms that use is whenever they have the template, a new nucleotide comes in and gets complementary, aligns itself complementary to the DNA template. If it's the wrong nucleotide, it makes a conformational change at the active site, and what the polymerase does is it kicks out that nucleotide and tries again, right? So it's kind of a proofreading. But what if it actually didn't work and it actually entered the wrong nucleotide? It still has a chance to remove this. And the way that this can happen is that it recognizes that there is a wrong base incorrectly incorporated, and the protein goes through a conformational change, and what it does, it takes out that newly synthesized strand that's shown in red here, and it flips it out to another region of the polymerase. This polymerase region has an enzymatic activity, which is the exonuclease activity, which means chewing out from the end, right? And that exonuclease activity removes that wrong base, and that removal again leads to a conformational change that flips this newly synthesized strand back into spot, and everything is back into normal. The polymerase has a chance to add another base. So this is called the proofreading activity. So 99% of the problems in the synthesis of the DNA in our cells through the replication machinery are fixed this way. But even when at 99%, given that a polymerase can make a mistake 1 out of every 100 nucleotides, this has a potential of 120,000 mistakes per division. And even though you can fix 99% of them, you still have a pretty good number that needs to be fixed by the repair machinery. Okay? So the repair machinery that fixes the problems that are coming out of the DNA polymerase is called the mismatch excision repair. Okay? These mismatch excision repair machinery recognizes the mismatches that are done due to incorporation of the wrong base. So this is done by a number of enzymes that actually travel with the DNA polymerases during replication, and they have a mechanism to sense which one of the strands are the newly synthesized strand and which one of the strands is the parental template. Okay? So due to this ability to recognize the newly synthesized daughter strand, the mismatch repair enzymes recognize the mismatch, and they bring an MLH1 endonuclease and another protein, another endonuclease called PMS2. Endonucleases means that you could cut through the backbone, remember? You don't need the free end. And what they do is they cut the DNA on both ends of the, like a little bit offset from the mismatch site. Okay? So that is going to remove a certain length of DNA fragment from the DNA. So you introduce these cuts, and with the help of a DNA helicase and an exonuclease, you basically remove completely this little fragment. So then what you have is you have the DNA polymerase coming in and filling up this gap, and then the DNA ligation is going to ligate, restoring the original information. Any questions about mismatch repair? Okay. What's another type of DNA damage that's common? Another common damage is from the sun. So although this looks awesome to me right now, you probably would like to move that chair under the shade, because the UV radiation also introduces a lot of damage to your cells. Okay, one of the most typical damage that introduced by the UV irradiation is the chemical cross-linking of the thymine dimers that are the thymine bases right next to each other. So normally what you have here is the two thymine bases, and the UV radiation cross-links these two. When these two bases are cross-linked, what it does, it makes a little bulge on the DNA backbone. They're not supposed to be cross-linked to each other, so that it creates a little DNA shape change, okay? And that shape change is recognized by proteins that scan the genome looking for weird shapes in the DNA, okay? So this initial recognition of a thymine dimer is made by a couple of proteins called XPC and 23B. And when they recognize that there is a damage in the DNA, what they do is they open up the double helix and start recruiting the downstream proteins to help fix the problem. So what they do is they recruit a few proteins, one of it which is called TF2H. This is actually also a transcription factor. It's that is used for transcription, and it has the name H here. You can always remember it stands for having a helicase activity. And you know what helicase is, right? It opens up the two strands. and it's prone to a lot of bad jokes, like if I can't remember, just look up online. Something about unzipping jeans. So this helicase open up the two strands, right? So the next phase is that you have a couple of endonucleases, again, doing the similar thing. What they are doing is they're cutting the sugar phosphate backbone a little bit offset from the actual original bulge site, right? So when that happens, it's going to leave this thing out, and so you're going to have a gap in the DNA double strand. So this gap is, again, recognized by the DNA polymerase, which is going to be filled and then ligated at the ends by the DNA ligase. Again, you restore the original information. Does that make sense? And actually, it's amazing. I can't remember the number, but there are hundreds of proteins involved in this process. We're only showing you a few very critical ones that explains the biochemistry behind it. And a lot of these proteins, the genes encoding for these proteins were found from people who have high incidence of skin cancer. They have a lot of, they're prone to a lot of skin cancer because they cannot fix the damages that are introduced by UV. All right. So next we're going to talk about what happens when you have a double strand break. So double strand breaks can be repaired by two basic mechanisms. One of is homologous recombination. Okay. So sometimes when you have a double strand break, this is very similar to the homologous recombination that occurs during meiosis, which you learned a few lectures ago. I'm going to go a little bit faster on this. What happens is that you have a double strand break, and the ends are digested by exonucleases, leaving the 3' stranded, single-stranded ends. These 3' single-stranded ends look for homology at a homology template, and they invade the double strand, the DNA, at where they find homology, and that homology basically it invades these. After it invades these strands, it gets extended by the DNA. The ends get extended by the DNA polymerase, leading to a couple of holiday junctions. And these holiday junctions are formed due to ligating the ends of the extended single-stranded DNA. And once these holiday junctions are resolved, what you're going to have is you're going to have the correct information. You're going to have an intact double-stranded DNA on both the donor template, that means the template that were invaded, and the original chromosome that was broken by the double-stranded break. Okay. So, at what cell cycle stage can this type of repair likely to happen? Homologous recombination. At what cell cycle stage would you expect the double-stranded breaks to be repaired by homologous recombination, based on the information I just gave you? S, yes, because the single-strand ends are going to look for homology. And the easiest time to find homology and when the two sister chromatids are next to each other, right? So if this damage happens in G1 or some other stage where the homology finding is very difficult, you use another mechanism of double-strand break repair, which is prone to more problems. Okay, so before that, because the double-strand breaks through homologous recombination depends on the fact that the ends are going to look for homology, DNA repeats in our genome is a problem for this kind of repair. Okay? So we've learned that a large proportion of our genomes are made out of repeats, right? These are sequences exactly the same, repeated many, many times. What happens? What might happen? All right, let's imagine that you have two repeat sequences at two distant sites in the chromosome, okay? And let's imagine that one of these sites contained a double-stranded break. Now, what you're going to have is that this double-stranded break is going to look for homology, right? And these sequences are going to look for homology to be able to repair themselves. Because the repeats are usually can have very similar sequences, right? And the homology searching is looking for short regions of homology. Sometimes it can mistakenly find the homology somewhere else in the genome, but that's not somewhere else in the genome inappropriately. Now, there are a couple of mechanisms in which this could be prevented. So, if this site forms a double holidate junction and goes through homologous recombination, what can happen is that you can actually form a circle that will excise out. Because the homologous recombination, the way the holidate junctions can resolve, sometimes can give to a problem where the middle of the DNA is out. But good thing, the repeats are not under strong selection. That means repeats can accumulate lots of mutations with no problem. The repeat sequences are usually not exactly the same. They're very similar, but sometimes you have one or two mismatches in between two repeats. And what that results in is these little bulges, right? These little mismatches in the DNA during homologous recombination. And the homologous recombination machinery is very strict. It doesn't allow any partial credits. So what it does is if it sees these mismatches, it aborts the recombination process. So you basically do not have a huge excised fragment through that. Okay? So another way to prevent recombination, another mechanism that prevents recombination between repeats is chromatinization of the repeats. We've learned that a lot of the repeats in our genomes are heterochromatinized. That is, they have a compact heterochromatin structure, right? So we have the repeat regions are mostly heterochromatic. And some euchromatin regions have more open chromatin. So how can this heterochromatin prevent homologous recombination between repeats? So the repeats are mostly heterochromatic here. And we said that if there is a double-strand break in one of them, it's going to look for homology, and sometimes it mistakenly finds it in another repeat. But this doesn't usually happen because homologous recombination machinery is really strict, and it doesn't happen due to this property of the repeats. But how? Why does this property of the repeats prevent homologous recombination? Any ideas?
Is the DNA just not accessible?
Exactly. The DNA is not accessible. For homologous recombination to occur, you have to bring an exonuclease. You have to chew up the ends. You need to have a single-stranded end that's going to look for homology. If where you're looking for homology is all closed and not accessible, you're not going to find it, right? Because the homology looking goes through strand invasion. And you cannot, the nucleosomes and the way that they're compacted doesn't allow strand invasion to occur. All right, very good. All right, so what happens then? You have a double strand break and you cannot repair it through homologous recombination. Double strand breaks are bad. If that occurs during cell cycle, the cell actually stops everything. There are signals that recognize that there is a double strand break, and what it does, it stops cell cycle. So these are checkpoint mechanisms. Checkpoints means there is a damage, you cannot go on without you fix it, because if you do, chromosome segregation will occur incorrectly, right? So there is another mechanism that is basically fail-safe, that is going to fix the double strand break no matter what, but it might make mistakes in the meantime. And this what we call is the non-homologous end joining. What this does is it recognizes that there is a double strand break, and sometimes these breaks can occur blunt, it can leave blunt ends, that means a clean cut, right? Or sometimes they can leave damaged bases at the end, or even short stretches of bases that do not have any homology, okay, that lack the counterpart. So what the homologous, non-homologous end-joining machinery does, which is has a DNA, the protein's DNA PK and Ku7080 heterodimer, is they recognize these double-strand breaks, they stick them together, they clean these ends by removing the damaged bases or these bases that are that do not have any counterpart. And through some other proteins that are not listed here, they directly ligate these ends. So in this example, we start with this here, and we go through the repair process. What kind of mutation would this picture, the mechanism in this picture would result in? What kind of mutation would happen at the end here? Deletion, right? Because that these original bases are removed here, this non-homologous end joining is going to introduce deletion. That doesn't always happen. If there is a blunt end, sometimes that will lead to a correct repair. But if there are sometimes damages, it's not going to lead to correct repair. That's why this non-homologous end joining has low fidelity. It's not very good. All right. So we said that the replication errors can cause both nicks and double-strand breaks in the DNA. We talked about how the nicks are going to be repaired by homologous recombination, right? But sometimes you have double-strand breaks as well. So which of these are more likely to cause genome rearrangements? Nick or a double-strand break? Which one is, in your opinion, would be more dangerous for the cell? Nick? Double-strand break. Because nicks are usually repaired by homologous recombination, and it's going to have a very high fidelity. The double-strand breaks can be repaired by non-homologous end joining, and end joining doesn't care where it's actually repairing to. Imagine that you have multiple double-strand breaks in the cell, right? And if the end joining machinery finds one end of the one chromosome and the other end of another chromosome that has double-strand breaks, sometimes it joins them, right? So the non-homologous recombination, non-homologous end joining only cares about joining the ends. It doesn't care what ends it's joining, right? So what that can cause is it can cause chromosomal translocations and all kinds of weird type of rearrangements in the genome. All right. Here's an example of such rearrangement that occur in chronic myelogenous leukemia. So what you have here is you have a famous chromosome called Philadelphia chromosome that it rises from a translocation between chromosome 3 and chromosome 9. And what happens is that this part of chromosome 9 gets fused to this part of chromosome 22, and this part of chromosome 22 gets translocated, that means fused to the end of chromosome 9. And how are these things detected? What can you do to detect these kind of rearrangements? What technique can you use? Fish and JAMSA staining, right? A classical way to look at cytogenetics. So, so here's a fish experiment. What you have here is you have the chromosome 9 labeled with a mixture of red probes, and chromosome 22 that is labeled with a bunch of green probes. So what you can see is the normal chromosome 9 in this person is completely red, and normal chromosome 22 is green. And what you can see here is that this person is carrying a chromosome where part of chromosome 22 is now fused to chromosome 9, giving this kind of pattern, right? And here's the Philadelphia chromosome, which is tiny and contains sequences from chromosome 9. All right. Because of these kind of arrangements, because of the damage that it can make into the cell, if you have mutations in your DNA repair enzymes, then you would be more prone to cancer and other hereditary diseases. The reason for that is we have an assault on our DNA all the time, and our DNA repair machineries are cleaning them up. If you don't clean, then those mutations are going to stay in the cells, and that can cause disease. So here is a table where a number of human hereditary diseases and cancers that are associated with DNA repair defects. In fact, a lot of these DNA repair genes have been isolated through these instances. So, for example, we talked about this skin cancer, which is caused by mutations in the nucleotide excision repair enzymes. So if these mutations are inherited from the parents, for example, in the case of breast cancer deficiency, BRCA1, BRCA2, which are involved in homologous recombination repair, this can cause a susceptibility to breast and ovarian cancer, right? Sometimes these mutant copies of the repair genes are inherited in one copy, right? So sometimes you have only one copy of the gene, the normal copy and the mutant copy. But because the DNA damage happens all the time, if by accident the other copy gets mutated and doesn't get repaired because there's a suboptimal amount of repair going on, now you have two copies mutated, right? And those cells will not be able to repair their DNA even less. They're going to be able to repair even less, and that's going to cause lots of extra mutations. All right. So that means that a lot of the tumor suppressors are actually DNA repair genes, and you can see here the DNA repair genes can be tumor suppressors. Some other genes who are not DNA repair proteins but tumor suppressors, that means genes that are important for not having like uncontrolled division, right? So these tumor suppressors, if they by accident get mutated because there is not enough DNA repair or there's a lot of damage that completely overwhelms the DNA repair mechanisms, then again that can cause cancer. All right. The link between DNA damaging agents and cancer has been recognized many years ago. Actually, it's been more than 200 years when people recognized that if there is a lot of smoke in the environment, that those people tend to get lung cancers, right? So a correlation can be made between the lung cancer and smoking many years ago. So here's a plot from your book where you can see along the axis of the time you have the cigarette consumption on the y-axis on the left, and the age-adjusted cancer death that is plotted on the y-axis on the right. And what you can see is that with increased cigarette consumption, you see an increased amount of lung cancer-related death in both males and females. So for many years, lung cancer was then correlated with smoking, but correlation is not causation, right? And a very good example of that is if you plot the number of pirates against global warming, you're going to see a decline, right? But that doesn't mean that global warming is reducing the number of pirates. These are correlations that you can make. That is not giving you any causation. But correlations are a good place to start, right? So if you notice that such correlations, you can look for causation. So when people recognize that the cigarette smoke can cause cancer, they said, okay, let's go and find out if this is causative. All right. So that direct link was then established through research. So the first observation that was that a lot of these lung cancers, so about 60% of these cancers contained a mutation in a tumor suppressor gene called P53. It's a very famous tumor suppressor that is mutated on a whole range of different cancers, including the lung cancer. All right. So when people looked at where were these mutations, right? So These mutations were found in codon amino acid coordinates 175, 248, and 273. Okay, so the researchers also knew that one of the candidate molecules in cigarette smoke and other types of smokes actually is a molecule called benzopyrene. So when this gets, there are some lung enzymes, there are some enzymes in the lung cells that can convert this benzopyrene to a carcinogen. That carcinogen then reacts with the DNA and basically adds a big adduct, right? Is a big molecule at a certain position in the DNA that causes a problem. So what kind of repair mechanisms can actually repair this when you have a big adduct on the DNA? It's modifying the base. Base excision repair, right? But if it doesn't get repaired, it can cause a mutation. All right. So what the researchers did next is they mixed bronchial cells with this chemical, benzopyrene, in a dish. And they said, let's see what kind of mutations are we going to get in the p53 genes after this incubation. Okay? So then they sequence the p53 gene, and what they find is they find exactly the same mutations at the codons 175, 248, and 273. So basically this carcinogen left its own footprint in the DNA that was also seen in the lung cancer patients. So the researchers then made a direct link in the ability of this agent to cause the exact same mutations in p53. And these mutations are not typically seen in other cancers that happen in other regions of the humans, like colon cancer and etc. So they can say, okay, this is really the smoking gun that's coming from the cigarette smoke into the lungs. All right. So this type of tissue-specific carcinogens is seen in other type of mechanisms as well. For example, here is a chemical called aflatoxin B that is found in moldy grains, and etc. So when it gets into the liver, due to some enzymes that are present in the liver doing some metabolic activities that is specific to the liver, these enzymes might also modify enzymatically this aflatoxin B. Again, what it does is it becomes a chemical adduct that can react and change the base in the DNA. Okay, and if this base is not repaired, again, it can cause mutations. So you would usually not like to eat anything with mold, but you might like a nice, well-cooked steak, and so that doesn't help either, because a lot of the meats that are cooked at high temperature contain this compound called PHIP. It's found in human diet in many cases, but grilling is one of the bad things. So what happens is that this gets, through the metabolic enzymes, get changed into a carcinogen, and that gets incorporated into the DNA as in the form of a large chemical adduct. All right. So we talk about mutations being all bad, right? But not all mutations are bad. A change in the DNA sequence can cause a problem, right? It can cause an amino acid sequence change in the protein, and that may affect the protein function. But sometimes it affects the protein function in a favorable way, given some certain circumstances. So if DNA repair were perfect, right, there would be no mutations in a population, and there would be no genetic variation. And that's not good. We want to be different from each other. So without genetic variation, then natural selection would not happen. So natural selection selects for differences that gives them some kind of advantage, right? So we should then have enough mistakes in a population to allow the evolutionary mechanisms to work. Just to basically tell you this, let's say these are individuals in the original population, and being blue is awesome, but then there is a condition that kills all the blue individuals, right? So what might happen is that if there is a mutation that arises in the population, giving rise to an orange guy, and this orange guy, oh my God, Trump. Okay, I should change this color. The orange guy survives. Oh my God. Okay, so this orange guy survives. What you can have is that the orange guys would divide and basically selected for this new environment, and the population is going to be made up of mostly orange guys. But then there might be another selection that happens, and then that gives rise to a green guy, and then that green guy gets the advantage, etc., giving rise to this very large genetic variability in the populations, right? All right, that's all good. That kind of framework in how the evolutionary mechanism works can be applied to a lot of different biological systems. One of which is the immune system. Okay, in immune system there is a theory called clonal selection theory. So what that means is that in our immune cells there are a whole bunch of different kinds of immune cells that are expressing receptors that can recognize a whole bunch of different antigens, right? So there's millions of different types of immune cells that can recognize millions of antigens. The reason why we want to recognize this antigen is that if we have parasites or something that is invading our bodies, we want to be able to start our immune system and attack it. But the first step of activating the immune system is the recognition of the antigen through binding, protein-protein binding to these receptors, okay? So what you do is you have a whole collection of antigens, antigen-specific receptors ready to go. All right, let's say there was an infection and a certain antigen that recognizes, that can bind to this receptor, binds to it. What it does is it activates the cell to divide. Now you make many of these immune cells that's going to produce lots of antibodies against this antigen, and the immune system is going to attack these antigens and clear whatever the infection is there, right? Right. I said that there were millions of different receptors. How many genes does a human genome have? 25,000, give or take, right? So if this was a pre-done system to make millions of different receptors, you would need 1 million genes, right? Which we clearly don't have. So how do we go about in generating this diversity? So, like I said, it's difficult to include the DNA sequences that would encode for this incredible diverse set of antibody repertoire. So that necessitates mechanisms to increase diversity. And one of these mechanisms I think you went through is the VDJ recombination. So basically using this combinatorial system to generate many different combinations of the antibodies. So you have several numbers of each segments of an antibody, and they can get combinatorially joined to each other, giving rise to many antibodies. However, this is not enough. So there's a second mechanism that purposely introduces mutations to antibodies to generate, introduces mutations to the genes that encode for the antibodies to generate these different receptors. Does this make sense so far? All right. So these this is called somatic hypermutation, and what this does is it helps to select for antibodies that have a higher affinity to a certain antigen. Okay? So once an antigen is recognized a little bit, just a little bit, so the binding is there, but not quite enough, and those cells get proliferated, right? So you have lots of those cells that are encoding for the antibody that kind of crappily recognize the antigen. Now what you want to do is you want to create better antibodies that can better bind to the antigen. So you want to increase the affinity of the antibody to the antigen. And that happens through affinity maturation that uses somatic hypermutation. Just to remind you how antibodies look like, right? So you have a heavy chain and a light chain, and you have an antigen that is bound to this surface. Now, this surface is called the complementary determining region because it binds to the antigen. And the shape of this surface, the changes in the shape of this surface is going to increase or decrease the affinity to the antigen. Okay? So what happens is that after the cells, the immune cells are recognized and proliferated, they induce an enzyme called activation-induced deaminase, AID. And what this enzyme does is it purposefully takes the methylated cytosine residues and it deaminates to produce T. Now, in these cells, they replicate fast and make lots of mutations. And these mutations can happen at all kinds of regions of the genome. So, but if it happens in the complementary region that binds to the antigen, it's going to make some changes, right? All right. So the B cells activate this enzyme that purposefully puts mutations. What would be the consequence of hypermutation? Hyper means lots, right? Lots of mutation. Hypermutation in most of the B cells. What's going to happen? You're purposefully making mutations. What could be consequence of making lots of mutations?
Like, you have a higher rate of making mutations, you can make a mutation in a state where you don't want to have one.
Yeah, you can make a mistake. You make a mutation in DNA polymerase. What's going to happen? You're not going to replicate. No good. You're dead, right? So in fact, a lot of these mutations can cause dead cells because they're no good. But what if, by chance, you made a mutation at the complementary region that changed the amino acid at that complementary region? So what could be different consequences of such a mutation at the complementary region that's binding to the antigen?
Better or worse binding?
Better or worse binding? What if it's better? What's going to happen to that cell that has that mutation, that binds even better? It's going to survive and it's going to actually get activated because it binds so much. Again, clonal theory, right? It activates and it gets replicated and it makes more and more. And that's one of the reasons why that there is a gradual increase to affinity to antigen across time. So after vaccination, you could take a while to make lots of these antibodies that are binding much better to the antigen, which is fascinating. You make you produce millions of cells, randomly mutagenize it, and select the one that works, right? That's exactly how kind of the evolutionary changes happen. All right. And that's kind of evolutionary concepts, right? This is the concepts that I'm trying to teach, is also can be applied to cancer. So each cancer cell basically accumulates mutations, and some of these mutations will be bad, so they're going to die, but some of them will be advantageous for growth. So this single cell that has a mutation that has advantage to grow will divide and increase in numbers, right, within the tumor until it accumulates more mutations that will give it more advantage, or another cell even with even better advantage can start to take over the tumor. So that's one of the reasons why you will hear that a lot of tumors that are isolated, you will hear a terminology, it'll say it's heterogeneous. That means there are lots of different cells in that tumor because they're doing all kinds of different things. What could be some of the mutations that can give a tumor cell advantage, right? So what can you have this advantage? One of the mutations that can happen in a tumor cell that is going to give it advantage is the inappropriate expression of the telomerase. Okay, so in normal cells that are embryonic, so they need to go through many divisions, or they're stem cells. They have the capability of going lots of divisions. What you have is you have lots of replication. I wouldn't call it indefinite. I don't know if anyone can do an experiment to see if a cell can indefinitely divide. Whoever is going to do that needs to live indefinitely, right? So that's another very conclusive experiment. But they can go on very long time. And one of the reasons they can do that is they have long telomeres, right? So in a somatic cell, so that's not a germ cell, like our skin cells or muscle cells, etc., the number of replications that a cell can make is limited because every successive division causes a shorter and shorter telomere. All right, so a cell without no telomere at the end is not able to divide anymore, because if they divide, what happens is the cell recognizes the ends as a break rather than the special telomere structure which requires those repeats, right? And when there's a break, it's going to stop or it's going to join. But telomeres has special mechanisms so that they don't get they don't get non-homologous end joining. So you don't end up like gluing all your genome. All right, but in cancer cells, what happens is that the telomerase is active, so you have very long telomeres, and this gives them the advantage of persistent growth, even though they are supposed to stop. But sometimes this can also lead to other problems. How does this work? Why do telomeres shorten in every replication cycle? Did you guys see this slide? You saw it, right? Okay, so I can go through it very quickly. So the reason for why that happens is that the lagging strands use an RNA primer to be able to replicate it in the reverse direction, right? So what happens is that in the last lagging strand replication, the primer is removed. So this end of the newly synthesized chromosome is shortened, and through new replications, that chromosome is going to get shorter and shorter and shorter. The telomerase, the complex is a complex of proteins that also contains a non-coding RNA that has a function. So what this non-coding RNA does is that this telomerase complex recognizes the ends of the DNA, the chromosomes, and uses this RNA template to synthesize DNA, new DNA. So what this can do is that the synthesis goes from this end and continues. And once it's done synthesizing this whole thing, it slides a little bit far off, and again uses and synthesizes new DNA. So successively increasing the length of the telomere, right? Here's a question for you. What enzymatic activity is in the telomerase? We've learned it in the transposon lecture. It is synthesizing DNA using RNA. Speak up. Reverse transcriptase. Transcription is going from DNA to RNA. It's doing the reverse, RNA to DNA, right? So it contains a reverse transcriptase enzyme. All right. So the cancer cells then have the ability to divide almost indefinitely, right? What's the evidence for that? The evidence is that there are cells that were isolated many years ago that they are still dividing. One of the most famous cell lines that has been used widely across many labs in the world, it was isolated from a woman at Johns Hopkins in the 50s named Henrietta Lacks. Okay? And these cells are named HeLa cells. they can divide indefinitely, and here's a picture of that because there are a lot of labs that are continuously culturing these cells. All right, some quick facts. Since its isolation, these were the first cells that were able to grow in dishes from isolated from a human. Back then, there was a lot of effort to isolate human cells and be able to grow it in the dishes because you can't do experiments in humans, right? You want to do experiments with using human cells in tissue culture. So the amount of HeLa cells that are grown across the world are estimated to be 20 tons. There are about 300 papers per month published using HeLa cells and refer to these cells. And in fact, Henrietta Lacks actually died quite young because of her cancer, and these cells lived outside Henrietta Lacks longer than she did. So these were isolated from the cancer cells in Henrietta. And there's actually an excellent book. It's called The Immortal Life of Henrietta Lacks that was published, and this is her family. It's a good read. All right. So how does the Henrietta Lacks cells look now, right? So there are HeLa cells that are distributed across the world, and if you karyotype these cells, what you see is there has been a lot of changes. And this is very typical for cancer cells. What you're going to see is here's a normal karyotype. This is the male karyotype. You have each of the chromosome, etc. And there are 23 pairs of it. If you look at the HeLa cells that was isolated from one of the labs, right? Even between labs there are differences. What you see is that these cells contain improper numbers of chromosomes. For example, it has 3 copies of chromosome 1, 1 copy of chromosome 2, 1 copy of chromosome 3, etc. And on top of it, it has a lot of chromosomes that were translocated, right? So you have chromosomes that are completely jumbled with each other. So the idea that people have now, which is not proven, is that some of these changes are not making any difference for the cells. So the cells have high amount of damage and jumbling, which we call genome instability. It has high genome instability, and some of these instability, the changes are not making any contribution. But some of them are advantageous. That means it's going to give advantage to that cell to be able to proliferate constantly and without control, right? So you can think of these cells basically evolutionarily adapting to growing in tissue culture media, tissue culture conditions, right? So these cells are going through these changes to adapt that. Okay, so that's it. Any questions? Go ahead.
DNA repair mechanisms have the ability to recognize the difference between introns and exons that might lead to differences in efficiency?
There are definitely differences between the amount of repair that occur in the transcribed regions versus non-transcribed regions. Guys, just hang in because the lecture is not done anyway. See, we're at 1 hour. We can sit another 10 minutes. But so we talked about the mismatch repair, right? Using a protein called TF2H. This is a transcription factor that typically binds to genes. And in that, it also uses its helicase activity to unzip the DNA so the transcription can go on, right? And you might think that this gene, this protein, is going to be found at mostly coding regions of the genome, right? So, because of this, the mismatch repair could be more active at coding regions of the genome, which is indeed the case. That's called transcription-coupled DNA repair. There are some other mechanisms and differences in the type of, in the regions of the genome and how they get repaired, but we don't know the details about them so much. Any other question? All right, let's get out.